I call this one the Campfire Song Song. Let's gather around the campfire and sing our campfire song. Our CAMPFIRESONG song. And if you don't think that we can sing it faster, then you're wrong. But it'll help if you just sing along. Certified
Alright, so you got some SpongeBob trap mix there. Believe it or not, this is the third attempt at recording this video because YouTube keeps jacking up my videos. So, let's try this just one more time. We previously talked about the two other layers of the eye. We talked about the fibrous tunic and we talked about the vascular tunic. Now we're getting into the third one that actually has to do with the generation of nerve impulses so that you actually have the ability to see. There are two parts to the retina or neural tunic. You have the pigmented part. The pigmented part's sole job is to absorb light rays and change light into a chemical substance. That chemical substance then triggers or stimulates what is called the neural part and specific cells called photoreceptors. The photoreceptors are divided into two categories based on how they look or their shape. You have rods and cones. But once the pigmented part absorbs that light and changes it into a chemical, that chemical stimulates the photoreceptors, which their sole job is to generate action potentials. That action potential then travels to your brain. But we're going to get into more depth with that here in a second. I want to show you this diagram. I'm going to draw on it because drawing is good. As the light passes through the cornea, passes through the pupil of the iris, and then gets refracted by the lens onto the retina itself, that light gets changed by the retina. So we're going to magnify this section of the retina here. And what you end up with is you end up with a complex system of cells, photoreceptors, and other specialized cells solely designated for the absorption of light. The first layer that I need you to realize is going to be this part right here. This is the pigment part. What light does is it comes through and passes through all of these cells and hits this pigmented portion. The pigmented portion absorbs that light and changes into chemical form. Now, once it changes into chemical form and that light passes through and hits that pigmented portion, changes into chemical form, these cones and rods in the neural section are stimulated, just the rods and cones right here, are stimulated by the chemical change and generate action potentials. These two layers of cells are ancillary or supporting cells that are part of the retina and the neural layer, and once they generate those action potentials, they converge into one portion in which they travel down to the optic disc, which is converge on the optic nerve and get sent to your brain. Like I was saying a second ago, the neural part has two main kinds of photoreceptors that I want you to concentrate on. They are rods and cones. So after that light is absorbed by the pigment layer and changed into chemical form, it actually runs and stimulates those rods and cones and actually causes you to be able to see eventually. But the two types, rods don't discriminate color. They're basically used for low light situations, and if your rods are being stimulated and they were the only thing being stimulated by the pigmented layer, then you would only be seeing in black and white. So imagine that you have the absence of cones in your retina. What that means is you don't see in color, you see in black and white. Cones, on the other hand, provide your color vision, and it's based on three different types of cones that are dedicated towards three different colors. We'll get into those colors in a second, but I want you to think about if you've ever had to change the cartridges on your printer, you will notice that there's a black cartridge, and then there are three colors. Those three colors are used in mixes in different amounts to generate all the different color spectrums that your color printer has the ability to print. I do want to show you some videos, though, before we go any further. And two of them are on the retinal organization. The first one's going to go over generalized the eye itself. The second one right here is going to go over the retina being divided into the pigment layer and the neural layer. And the third one's going to be a video of the Blue Man Group that I just find very interesting.
Light rays enter the eye through the cornea, pupil, and lens. These light rays pass through the vitreous, a clear gel-like substance that fills the middle of the eye. The light rays are focused on the retina, a light-sensitive tissue lining the back of the eye. The macula is a very small area at the center of the retina that gives us our fine pinpoint central vision. The area of retina surrounding the macula gives us our peripheral or side vision. The retina converts the light rays into signals that are sent through the optic nerve to the brain.
Okay, so that one just basically goes over the generalized structure of the eye and the retina and how it goes from light and being projected onto the retina itself. What we're going to get into now is the actual structure of the retinal layer.
The human eye is a complex organ that provides visual sensory information to the brain. Each part of the eye performs a specific function. The retina is responsible for receiving light information. Its pigmented layer helps to capture light. The retina's sensory layer has rod and cone photoreceptors that detect and translate light information into nerve impulses. The signal is passed through the retina's layers of neurons and carried to the optic nerve located at the back of the eye, where the signal is gathered and sent to the brain.
So in that video, I hope you noticed that, if I pause it real quick, you'll notice that these are all the nerves, or I'm sorry, not nerves, neurons that are attached to the neural layer of the eye. So from this area here down to this area here, the green and blue, the brownish color and the yellow, all part of the neural layer. This is your pigment layer. So they showed you in a very great way that the light actually passes through all of this layer, and which is absorbed by the pigmented layer, changed into a chemical signal, which changes into an electrical or action potential signal generated by the cones in the rods that then travel to the optic nerve. Now, like I said, I do want to show you one video that I think it's pretty neat. If you've ever seen the Blue Man Group, it is actually relevant to this class because they do give great anatomical, physiological information, but in an entertaining form. So we'll just watch it for fun.
Each of your eyes has over 3 million photoreceptors called rods and cones. These receptors convert light into electrochemical signals that travel through the optic nerve and into the brain. Here, these signals trigger the neurological process scientists call the Hemmelreich Shiznit that happens inside your bristle. In your eyes specialize in night vision and can function at much lower light levels than cones, but they do not respond to color. This is why we can only see in black and white when we are in the dark. The cones in your eyes, on the other hand, specialize in color and come in 3 types. Those that respond to the color red, those that respond to the color green, and those that respond to the color blue. All of the colors that we experience are the result of these 3 types of cones firing off in different combinations. When you look at something, the rods and cones in your eyes fire in rapid succession. But between each firing, there is a brief resetting period during which your eyes are unable to take in any new information. Your brain covers up these microscopic moments of blindness with lingering after images which help make your vision appear to be fluid and uninterrupted, even though it is not. This phenomenon, known as persistence of vision, is the unique physiological quirk that makes the illusion of animation possible. The dark spaces between each still frame of animation literally sneak by while your eyes are not looking.
Was a little long and took up a portion of your time, but I just find it really interesting because they use highly visually stimulating theatrics to actually present you with visual information. So, like I said earlier, there are 3 different types of cones. Remember, 2 photoreceptors, 3 different kinds of cones. But you have blue, red, and green, so think of those like in different combinations. They fire off in different combinations to produce the different color, the various color spectrum that the human eye can see. Color blindness, a sex-linked chromosomal disorder, actually occurs when one or more of the types of cones are absent in the eye or they do not function properly. And what they do is a very simple test called the Ishihara test to determine if somebody actually is colorblind. What it consists of are several cards or plates that look like this. There's a colored background, and then there's a number embedded within the actual plate that reads in a different color. When somebody is colorblind, they will not be able to see either the number itself or the color in the background. So in this particular instance here, it's a red 66. In this example right here, you have a red 42 on a green background. And then this one here, well, that's the one we just did. And they will rate the ability to see or not to see all those different things and determine color blindness. And I opened up a bunch of those. There are other cells within the neural layer called bipolar and ganglion cells that actually support the function of rods and cones in generating the action potentials. Not necessary for you to know a whole lot about them besides the fact that they exist, and they're the end point before it turns into the nerve fibers or neurons that are going to converge on the optic nerve. Pigmented layer, rods and cones, all of this is part of the neural layer, the pigmented layer. The macula, lutea, and fovea of the retina is the focal point of the retina where light images are projected by the lens. You want the light projected by the lens to hit these points. This is what gives us our fine vision of what we're reading and seeing directly in front of us. When that is not projected on there, or that light is spread across the retina, anything that is not projected on the macula, lutea, or fovea is actually called our peripheral vision. That's why our peripheral vision isn't as accurate or as fine-tuned is because there's a higher concentration of photoreceptors at the macula, lutea, and fovea. When you have nearsightedness or farsightedness, that is where the lens projects the image either too far behind the macula, lutea, or fovea, or in front of. And that's based on the depth of the eye, the roundness of the eye, or whether there's astigmatisms. All those different things cause the lens to project it in a wrong place, which produces blurred or misconceived vision. Now, let's talk about the optic disc. I think it's actually better just to have a diagram first, but what you'll see is your retina is all of this layer here and all of this layer here, and it kind of surrounds the eye. But you'll notice there's a break in the retinal layer right here that is composed of all the neurons converging on one spot to become the optic nerve. That one spot is called the optic disc. That optic disc is in fact your blind spot. So whenever light is actually projected by the lens on that one spot, you end up with a lack of vision in that area. We did this when we did the cross and dot experiment in our visions test and demonstrations lab, where you slid the card that had the cross and the dot on the outside, and as you slid it closer and closer to your face, at one point, usually around 6 inches or less from your face, that dot will disappear. And you have to move it slow in order to see it, and then all of a sudden, after just moving it a little bit, it will reappear in your vision. That's because the light has been projected and passed over the optic disc, which eliminates your ability to actually see that dot. So the following is what we're going to go over in terms of the neural pathway of how it goes from light all the way to a signal in your brain. So before anything occurs, light in the environment has to pass through the cornea, which passes through the iris, and the hole in the iris called the pupil, and the pupil changes the amount of light that will be allowed to hit the lens. The lens then projects or focuses all of that light directly on the retina, particularly the fovea. Once that light hits the retina and passes through the neural layer on its way to the pigment layer, that pigment layer changes it from light into a chemical form, and that chemical form is used to stimulate the rods and cones or photoreceptors. The light information from that has now been stimulated or received into the rods or cones is changed into electrical impulses or action potentials that are going to travel through the rods and cones, followed by the bipolar cells, followed by the ganglion cells, until they start ending up in the nerve fibers. Once they're in the nerve fibers, every nerve fiber then converges onto one spot called the optic disc, and all of those neurons and nerve fibers turn into the optic nerve. You actually have two optic nerves. This should seem obvious because you have two eyes, unless of course you're missing an eye. Once that action potential is converged upon the optic nerve, it travels through the optic nerves into your skull. Well, actually it's already in the skull, but where it travels to the optic chiasm, where those two optic nerves cross and switch to opposite sides of the brain. Then they enter the brain, they go to the diencephalon. From the diencephalon, they're routed to the thalamus. The thalamus is in fact like the switchboard. It then takes that signal and says, okay, we need to send some of this to the visual cortex, we need to send some of this to the Broca's area, we need to send some of this to the Wernicke's area, we need to send some of this to the sensory motor cortex. All of these different areas, your thalamus routes it there, because not everything needs to go directly to the visual cortex for interpretation. It actually can go other places also. And those two main places are going to be the visual cortex, which is in the occipital lobes. Here are some other videos for you to watch if you feel like it. They're in the PowerPoints that I posted onto Schoology. Other than that, as always, if you have any questions, let me know.